Hi, I'm going to talk about dihybrid crosses. Dihybrid crosses is going to be just an examination of two traits. So, when we use dihybrid crosses, it is simple dominance, but now you're using two traits or four letters. Di means two. So you're going to examine two single separate traits in a single cross. Now I'm going to use Gregor Mendel's plants as my examples. So when we do the study of pea plants, if we said that round seeds are true breeding, we could say big R, little r, big R are round seeds. This is a true breeding strain. And your wrinkled seeds are recessive true breeding strains. You could cross a tall plant with round seeds with a wrinkled plant that has a short plant that has wrinkled seeds. So you're crossing two separate traits instead of one. All the other traits that we've done in the past is just one single trait. So if we basically crossed, we could say a round seed, tall plant, that's two single traits, times a wrinkled seed, little r, with a short plant. It would look like this. We're talking about two different traits. So the two different traits that I'm crossing is going to be seed shape. So round seeds versus wrinkled seeds. That's one trait. And the second trait that we're going to be discussing are tall plants versus short plants. Now we're using Gregor Mendel's method. So we start with an F1 generation. So your F1 generation, we learned that the F1 generation is when you cross two true breeding plants. So these would be two true breeding plants, for two traits. F1 generation is going to be the offspring produced by crossing two true breeding strains. So these are true breeding. Every time you have a round seed tall plant producing its own seeds, it produces plants that are round and tall. And every time you have a short plant with wrinkled seed pods, it's always going to be that. Now when you do a dihybrid cross, it looks a little bit different when you do a dihybrid cross because you have to have more squares to represent your principle of segregation when they separate. So we're going to talk about the F1 generation. So we're using Mendel's generation. You're crossing true breeding strains. So the F1 generation, again, is your first filial generation. We're going to be looking at the offspring produced by crossing two true breeding strains. But the difference between monohybrid and dihybrid is you're crossing two traits. So you can't use a Punnett square to easily show this because you're using two traits. So when we talk about true breeding and we cross them, it's super easy if it's a monohybrid cross because you're just going to use a normal Punnett square. So if we were just doing tall versus short, we would use a regular Punnett square. Now where this gets tricky in an F1 generation is understanding how to write out your information on how they separate. So first thing you need to do is you need to draw, it's a Punnett square, but instead it has 16 squares. All right, so when we have the principle of segregation, the alleles have to separate. Now, the way that you do this is you place your one plant, which is homozygous dominant for round seeds and homozygous dominant for tall seeds, and those will be on the top. And then you take your homozygous recessive wrinkled seed and your homozygous short plant, and you'll put this on the side. Now, there's a way that you have to write this out. The first thing that we're gonna always do is what we call like the FOIL method. The first R during the principle of segregation can attach to the first dominant till T. So R, T, that's what you put on the first square. Now, principal segregation says that this R can also randomly pair with this allele. So you have the first one and the second one. So you have another big R and a big T. So you've taken care of the first R. So then you take care of the second R. The first R or the second R can bind randomly with the first T, or it can randomly bind with the fourth one, so you get RT. So this is the principle of segregation, that our alleles are going to segregate. That is how you figure out what is written on the top for the first plant. Now you do the same thing for your homozygous recessive wrinkled short plant. So the first R can randomly assort with the little t, and then the second R can randomly assort with the next little t. 1 and 2. So you have R little t, R little t, and you do the same thing on the bottom. So 1, 2, and then you go to the second R, 3, and 4. So you can get little r, little t, little r, little t. Now this is how you set up your cross. Now when you're doing a dihybrid cross, you always keep your R's together and your T's together to figure out your first generation, just like we have here. The R's stay together and the T's stay together. So when you're doing your cross, you do one square at a time. So in this first square, you would get a big R, because you always put your dominant trait first, and then your little r, and then your big T, and then your little t. So that's the first square. Now, when talking about this offspring, we know that this is simple dominant, so it has a big R, so it's going to have round seeds. So it has a round seed because round, this big R is dominant, and it's tall because it has a dominant trait. So this is a round seed tall plant. Now you do these for each square. Now because all of these are the same and all of these are the same, then all of these are going to end up the same as well. So you have your big R, big T, little t. So this is its genotype. It's heterozygous round and heterozygous tall, but its phenotype is it's round and tall. Big R, big T, little t. Big R, big T, little t. Now you would do this and these would be the same all the way throughout. So I'm not going to finish this. But when we talked about the F1 generation, all of the offspring ended up having the same genotype. So actually I'm going to fill this in. Big R, big T, little t. This is your F1 generation. I would just go through big R, big T, little t, big R, big T, little t. Since this is the F1 generation, we can say that all of the offspring have the same genotype and the same phenotype. So you need to know the words genotype and phenotype. So in your F1 generation, you had 16. Big R, little r means heterozygous. So they're heterozygous round and heterozygous tall plants because this is simple dominance. It is round and tall. So these are your genotypes. Your genotypes is heterozygous round, heterozygous tall plant. But you also have 16 round / tall plants. That is its phenotype, its physical appearance. So if we ask the genotypes, you would have to write out the individual ones. This is the F1 generation. All of the F1 generation have dominant phenotypes. They're round and tall. Now, when you say F2 generation, just like you had in simple dominance, the F2 generation, you have to cross or self-fertilize your generation 1. So you cross your F1 generation or self-fertilize. So we have to self-fertilize 2 of these and do a Punnett square. So F2 generation. But we're doing an F2 generation of a dihybrid cross. So F2 generation is going to be self-fertilization of offspring from the F1 generation. So we know what our cross is. We have a heterozygous round, heterozygous tall plant self-fertilized with itself. So we're still using a dihybrid cross or 2 traits. So we have to draw out a Punnett square with 16 squares. Okay, for the first plant, you do the FOIL method. It's a 1, 2, 3, 4. Big R can randomly join with the big T because of the principle of segmentation. The second one can bind with the little t. So you say big R, little t. Now you move to the second R. The little r can bind with the big T, so little r, big T. Third is the little r combined with the little t. Now, that is the first plant. Now you need to do the same thing with the second one. Big R, big T, big R, little t. Now you do the second R. Little r, big T, little r, little t. Now, where this gets confusing is students are not careful when they're doing their crosses. Remember, you have to go down and across. You have to keep your R's together and your T's together. So what I personally like to do is my R's and then I go and do my T's. So in this first square, you have one big R and a second big R. Then you come down. I like to do my R's first. A dominant big R, another dominant allele. Then you go to the next one, a big R, little r. Go to the next one, big R, little r. The next one, big R, big R. I always put my dominant allele in front. Now, if you come to a cross where there's 2 little r's, just put 2 little r's. All right, now that I'm done with that, I can go back and do my second trait. It's a dihybrid cross. So you can color code these, or you can keep it all one color. I sometimes like to color code it just so that it helps me, but I'm not going to. So big T, big T, big T, little t. Now, I would take your time in doing these because it's really easy to make a mistake, and make sure that you're writing clear because it'll help you when you're trying to understand your phenotypes. All right, now, these all have different genotypes. So when looking at the first square, we could say this is a homozygous round, homozygous tall plant. Go to the next one. Homozygous round, heterozygous tall. It's simple dominant, so the big T still trumps the little t. The next one, heterozygous round, homozygous tall plant. So you have to change out the words homozygous and heterozygous for each one. The fourth one, heterozygous round, heterozygous tall. The next one, homozygous round, heterozygous tall. Homozygous round, homozygous recessive, short. So when we do the phenotypes for the F2 generation, we have to understand what the dominant traits are. So we always start with round, this is the dominant trait, and tall. So you count the number of plants that are going to be round, because that's dominant, and tall. So we're doing phenotypic ratios. So I want to know how many round and tall. That's the first thing that you always figure out, because round is dominant and tall is dominant. So even if it is heterozygous, it is considered to be tall. Now, after you found out how many were round and tall, then you need to calculate the number that are round, but they are homozygous recessive, so they're short. This is going to be a number. Then you calculate not if they're round, but the next thing is if they're wrinkled. So wrinkled means they're homozygous recessive, 2 little r's, but they are tall, so their second trait is tall. And then the last thing that you would be calculating would be if they're wrinkled and short, which I ran out of room, so that would be the fourth thing that you would want to figure out right here. So if they're wrinkled and short, that means they're recessive for both traits. So you're always going to end up with 4 numbers to represent your phenotypic ratio. Now, it may be easier to basically, check these as you go. So you have to find something that's round and tall. It has to be homozygous dominant or heterozygous to be read. But it has to have a big R and a big T. So big R, big T, 1. Big R, big T, 2, 3, 4, 5, nope, 6, 7, 8, nope, 9, 9. So I have 9 that are round and tall because they have large R's or a large T. So you would write, I have 9 that are round and tall. So the next thing is I want to know if they're round and short, but the only way they're short is if they have 2 little t's. So they have to have a big R and 2 little t's. So big R, move this over a little bit. Big R's and little t's. 1. I don't go through the ones I've already checked off. Round, big R, and little t's. So big R, little t's. 2, no, no, yes. So there's 3 that are round and tall. Now wrinkled means that they have to have little r's. So I only have 4 squares left. So I'm only looking at the 4 that are left. So wrinkled means that they have little r's. And I want to know if they're tall, so they have to have big T's, whether they're homozygous dominant or heterozygous. So little r's, big T. 1, 2, 3. So I have 3 that are wrinkled and tall. And I only have 1 square that's left. Wrinkled means that they're recessive, and short means they're recessive. So they have little letters. I only have 1. So when you calculate these phenotypic ratios, to know that you did it right, since there's 16 squares, these numbers should equal 16. So 9, 3, 3, and 1. These are called your phenotypic ratios. 9, 10, 11, 12, 13, 14, 15, 16. You have a total of 16, and these are your phenotypes. So when we talk about the phenotypes, we would say the phenotypic ratio, the way that we did it would be 9, 3, 3, 1. That is your phenotypic ratio. So this is the F2 generation phenotypic ratio. So we crossed 2 of our F1 generation for a dihybrid cross to get a phenotypic ratio of 9, 3, 3, 1. That is the set ratio for a F2 generation. And that brings me to one more principle of Mendel, the principle of independent assortment. In a dihybrid cross, the alleles of each gene assort independently. Now, that is why we did your FOIL method. They have to randomly join. So the big R can randomly join with the big T or the little t. They assort independently. They do not stick with that gene, so you have variations. That's the principle of independent assortment. So if I asked you how many gametes have genotypes that are homozygous recessive for both traits, you would basically say 1. But for each one of these, you would have to go through the genotypes. Genotypes makes it much more confusing. This genotype is homozygous dominant round, homozygous dominant tall. So you have to represent them independently. And these also can assort independently. And that's why we call it the principle of independent assortment.